Sometimes you meet friends in life that you know somehow are the best ones you're ever going to have. Somehow you even know it when you first meet them. I just said goodbye to a friend like that who's been staying with me for 2 weeks. So. Good morning. It's Monday, September 30th in 2019. It's wet today. I drive a little more careful. So this paper is a very straightforward paper, unlike the previous 3 that you guys have covered. This paper has a lot less nuance and detail to it, and in fact is just a straight up who's connected to who kind of paper. And in a lot of ways I think this paper would have been more appropriate as a starting paper almost, you know. Could have started here and then went with Anne-Marie's paper and then went with the addiction circuit from last week. But here we are. This is a paper that discusses electron microscopy data. And electron microscopy is very, very cool because it is the gold standard in a lot of ways for structural biology. If you want to say that 2 structures are connected to one another and how they are connected, we know a great deal about how those connections should look ultrastructurally from an electron microscopic point of view. That means that there are aspects of the ultrastructure of synapses and of neurons that we have seen in different parts of the brain, and so we can look at the ultrastructure of connections between neurons if we can label them usefully, and then see whether the structures that belie connectivity are present. Electron microscopy allows you to label specific parts of tissue and label them in such a way so that you can look at, for example, cell specificity from target. So in this case, what they're going to do is put a marker in to the lateral habenula and label the neurons in the lateral habenula, and then these neurons will be labeled all the way to the VTA, where they will then look at where those axons that are labeled from injection in the lateral habenula, where those axons and who those axons are making contact with in the target structure, the VTA. So, just a few things. The VTA is the source of the dopamine signal in the brain. Dopamine is a reward signal, and dopamine has been studied extensively in drug abuse and reinforcement learning. So the dopamine signal from VTA is well understood in terms of how it correlates with behavior, and especially how it correlates with reward. And thus finding out how the dopaminergic neurons in the VTA are driven and who drives them during behavior is a pretty key question to understanding how dopamine can regulate behavior. So this paper then posits that because the lateral habenula shows opposite activity patterns to the VTA, and that it projects there, that there's a good hypothesis supported by that data which suggests that maybe the lateral habenula projects to local GABAergic cells in the VTA being made up of dopaminergic cells and GABAergic cells, that if you projected to the GABAergic local interneurons, you could turn off the dopaminergic cells in the VTA. And then this circuit wiring would then be sufficient to explain how the lateral habenula could control dopamine release via the VTA and the interneurons present there. So their hypothesis is that if they label lateral habenula neurons and look at their axons at the ultrastructural level in the VTA, they will see evidence at the ultrastructural level of synapses from the lateral habenula being formed on GABAergic interneurons. And so from the perspective of this study, then there are a few things that you need to know about what you can see in an electron micrograph. The first thing you can see is synapses close enough at a high enough magnification so that you can see whether they have an asymmetric or symmetric appearance. An asymmetric or asymmetric appearance of synapses tends to indicate an excitatory synapse because of the postsynaptic density being so well organized and full of proteins. So an asymmetric synapse at the electron microscopy level is thought to be a good indicator of an excitatory synapse. Asymmetric. Symmetric synapse, because the postsynaptic density is not so well organized, it just tends to look more symmetrical. And hence the names. So that's one of the things they could expect to see here, is the lateral habenula neurons have been presumed to make excitatory synapses in the VTA. And so one of the first things they want to look at is the lateral habenula positive labeled neurons and their axons in the VTA and what type of synapses they oppose. What type of synapses do they make? So if they made all asymmetric synapses, then we would be more or less confirming with this data that indeed they're excitatory synapses. But they do see both. And in figure one, they're very specifically looking at those terminals in the VTA and how they're labeled. They can see different combinations of asymmetric and symmetric synapses and lateral habenula and non-lateral habenula terminals. And that's important because they want to look at positive and negative terminals in this paper. Terminals that are positive for being in the lateral habenula and terminals that are not, coming from elsewhere. So they can use these as control group and experimental group if they're clever. So that's figure one. They're just looking at the axons in VTA and show you that they can label them, show you that they can identify synapses made by these terminals. And interestingly, in this one, I think they also show myelination and non-myelination, and that doesn't necessarily mean much anymore. It used to mean, actually figure two shows the myelination, I think. Myelination is also something you can see in the ultrastructure under an electron microscope. But myelination used to really mean excitatory axons, and now we know that that's not the case. A few papers have come out recently that have shown that interneurons are also myelinated. It's just some special types of interneurons only, and not all interneuron axons are labeled myelinated. But fast spiking cell axons are myelinated throughout mouse and human cortex. So it isn't ridiculous to think that in the past that this would have been a marker for excitatory synapses and excitatory axons, but nowadays I think that's too much to assume. But that's why they show it here, because they want to show you that they can look at axons and show that they are myelinated or not myelinated, because that can be some indication of excitatory projections. It's not the only indication. It's not a great one anymore. It's not the best one anymore, but it's not a bad one. As a first pass screen, is it excitatory or not? It's not a bad one. Figure 3 shows these same axons now opposing V-glut positive terminal, V-glut regions, V-glut positive, and V-glut is the abbreviation for the enzyme found in inhibitory neurons. It's all GABAergic interneurons, really. So it's not a very specific label, but it does label for inhibitory terminals and inhibitory dendrites, inhibitory tissue, let's say. So having that label there should provide a way to look at the fall positive terminals to see whether they are opposing GABAergic tissue, or in this case V-glut positive tissue. So it's just one marker for GABAergic tissue, which they think, or they hypothesize, could be the target of these lateral habenula neurons. I think I'm starting to grind metal in my front wheel. Got to change my brakes. So figure 4 is going to use post-embedding immunogold staining to show a similar deal, but now I think in figure 4, instead of doing V-glut staining, they're doing GABAergic staining with these gold particles. Is that right? Yes, immunogold labeling is done to show GABAergic tissue, GABAergic positive tissue. And again, this is just another way of labeling inhibitory neurons, another way of checking to what extent are these terminals coming from lateral habenula excitatory and synapsing on GABAergic VTA neurons. And in reality, it ain't very easy to show. Because they see both, right? They see GABAergic synapses, but they also see non-GABAergic or non-GABA labeled terminals, or non-GABA labeled tissue being synapsed upon by these lateral habenula terminals. So far, the paper's pretty straightforward, but figure 5 is pretty confusing to me. Figure 5, it requires you to pay attention to the n numbers that they describe. The n numbers that they describe in the figure legend. The gray bars are the total number of lateral habenula axons that they labeled. And then they break down those by symmetric and asymmetric synapses. And then they compare that to the non-labeled ones and how many of those were asymmetric or symmetric. And that's overlaid on the number of GABAergic particles. There were GABAergic gold particles. So what you see there is only 2 axons, one in the 40 to 60 group and one in the 100 to 120 group. Only 2 axons are positive for the fall staining, which is the indicator that they were in the lateral habenula in origin, and positive also for GABA. There are only 2. So of all the ones that they studied, 2 out of 26. So the vast majority are forming asymmetric synapses and are GABAergic negative. Remember that the particles show up everywhere, right? And that was also shown in figure 4, that when they immunostain with the gold particles for GABA, they get those particles to show up everywhere. It's like a uniform distribution, that then the uniform distribution shows up especially strong in the cells that are positive for GABA and less strong in the cells that are negative. And then it's the comparison of those that give you the cells that are positive for GABA. And so that means that all the cells will have some particles in them, but the ones that are on the far left of the scale are considered negative for GABA. And as you go to the right on the scale, then they're more positive for GABA. And there's only 2 axons that are on that side of the scale. Jesus Christ. I would like to say thank you or excuse me or sorry, but the youth of today, they don't do that. They have no need for sorry or excuse me. They just do their thing and wait for you to freak out. If you freak out, then they do their thing. So a few questions for the author, I guess, just to try and get you started on that. The first one would be, in order to do this experiment in a way that would actually answer the question functionally, we'd have to do it with a label that would allow us to test these synapses and how they work. So one way to do it would be to inject chanorodopsin into a virus carrying chanorodopsin into the lateral habenula, and instead of labeling and looking at ultrastructural changes or ultrastructural differences, you could cut slices and you could just stimulate the terminals labeled in lateral habenula, labeled in VTA, and record from neurons in the VTA to see who gets input. Now, this data set strongly suggests that both dopaminergic and GABAergic cells will get input, but they don't know that. And last week, you guys covered a phenomenon that could also be present here. Imagine a scenario where the excitatory synapses, the asymmetric synapses that are made on the non-GABAergic cells are silent synapses. Those silent synapses might only come alive after several times of no reward and needing to change the circuit in the VTA, so that after repetitive activation of these terminals, those silent synapses could become functional and then modulate what was a complete inhibitory signal at first to become a gated feed-forward inhibition signal that might allow the circuit to participate and to go through sculpting. And so you could ask them, one, are there specific markers for lateral habenula neurons that you could use to drive channelrhodopsin expression there virally, so that you could label the axons in VTA functionally and then stimulate and record and even see what they do in vivo. But I guess that's already been done. But you could ask that too. You could also ask, does the lateral habenula signal for negative consequences, negative reward, or only for the absence of a positive reward? These kinds of subtleties are very important in terms of dissecting valence in the brain. A great example of this would be the amygdala, which for a long time has been associated with fear conditioning, but more and more we're coming around to the idea that the amygdala might just be valence, positive or negative. And so as we sort these circuits out, the details about when these nuclei are active relative to other nuclei will be crucial to trying to draw inferences about how they affect behavior. I might just end it right here. That's pretty good, guys. I don't have much more to add. Like I said this in the morning, this is a straightforward paper. And it's a great one because it's largely a negative result. But it's a negative result that contributes positively to our model of this circuit. Unless you ignore this paper, you can no longer say that all the data indicates that lateral habenula turns off the VTA. Because it's not that simple. And that's why they show in figure 9 the idea that this changed their model. The hypothesis of the model is shown. I mean, he's in the middle. It's crazy. Hello, Rubius. Hello, Rubus, Bambubus, Patubus. Heb je pijn in je oren? Heb je pijn in je oren? Hello, Rubus, Bambubus, Patubus. Hey, buddy. Hey, buddy. Yeah, hey, buddy. Good to see you, buddy. Yeah, good to see you, buddy. Whoa.